Hello, welcome to your own channel, Curious Corners in Biology, where we discuss the curiosity of this subject of biology, where we discuss the unanswered questions in the literature of biology, unasked and unanswered questions. So, as usual, today we will be continuing with physiology of digestion. There are a few more questions. So, the first question, again, which I would like to discuss, is about how to detect lactose intolerance. So first let me share my screen. So as you can see my screen, how can hydrogen breath test aid in diagnosis of lactose intolerance? So we already know that lactose intolerance is the inability of a person to digest lactose into glucose and galactose. This is due to the deficiency of enzyme lactase. So, of course, the person has irritable bowels, abdominal cramps. These are certain symptoms of lactose intolerance. And together, it is also making up the irritable bowel syndrome, IBS. So, now let us understand what happens in a normal person and what happens in a lactose intolerant person. So, as you can see on the diagram on the left side, lactose, usually in presence of enzyme lactase, is digested into glucose and galactose. So normal stools are produced by normal metabolism. But when a person is lactose intolerant, we know that lactase is deficient. So lactose will not be digested into glucose and galactose in lactose intolerant person. So bacteria would ferment the lactose. So fermentation, we know, is a process that leads to production of gas and acids. So gases, especially hydrogen, is produced. There are, of course, other organic acids produced and other osmotically active molecules. Okay. There is also increased motility, which causes abdominal cramps and some discomfort in the abdomen. So here, when gases are produced, the major one of those gases is hydrogen. So hydrogen can be detected in the stools of lactose intolerant person, and this is how hydrogen breath test helps in diagnosis of lactose intolerance. Of course, the hydrogen also is let out through the breath, which is not usually the case with normal person. Okay. Now, the other interesting question is that we know pepsin and trypsin are the usual proteolytic enzymes of our gut, and they are capable of digesting dietary protein. Then why not keratin? Keratin is a protein which is usually present in our hairs and nails. Why so? Now, the reason is that pepsin and trypsin cannot break the strong disulfide linkages and hydrophobic interactions which are present in keratin. It holds the molecules of keratin very strongly. So these proteolytic enzymes are not sufficient to digest keratin. Then the question is, who digests keratin? Keratin is digested by enzymes keratinases, which are produced by certain groups of bacteria belonging to Bacillus species, certain groups of fungi like Microsporum, Chrysosporum. They are also known to be produced by insects like moths and beetles, and even vultures produce them. Keratinases. So those are the enzymes which will digest and hydrolyze keratin. Hydrolyze simply means the enzyme breaks down keratin in presence of water. Okay. The next question is very interesting. Often you might have heard that we should eat plant-based dietary fiber that is good for alimentary canal, that is good for gut health. But the question is how? What does it do? Is it only the soluble plant-based dietary fiber available? Is it only that, or are there other kind of dietary fibers based on plant available? Yes, there is also insoluble dietary fiber. Insoluble dietary fiber in plant-based diet would simply mean presence of cellulose fibers, pectin, lignin fibers. Now, these are passed through the alimentary canal unchanged, and they will have no effect on the digested food. What is important to understand is plant-based soluble fiber, which is dietary fiber. Now, soluble simply means that this plant-based fiber is able to dissolve in water. Okay. So, when it is present with food or partially digested food, what it does is it forms a gel-like structure. So, in a way, it hardens the consistency of the food, or we can say, in other words, the viscosity of the food is increased. Where do we get plant-based dietary soluble fibers? From vegetables and fruits like beans, broccoli, fruits like citrus fruits. Okay, these are high on plant-based dietary soluble fiber. Okay, now plant-based soluble fiber, as I mentioned, will slow down the motility of the food along the alimentary canal. So this will give more time for the enzymes to act upon the food which is present in the intestine. So the food will be completely digested, perfectly digested, and much time is also available for absorption of food. So in a way, the bowel transit time is slowed down. The bowel transit time is slowed down. Now, it is also understood that plant-based soluble dietary fiber also helps to reduce cholesterol in our body. So if it is reducing cholesterol, it will help a long way in managing health. It is known to reduce atherosclerosis. It is known to prevent diabetes. It is known to reduce obesity. Okay. All metabolic disorders are, the incidence will be minimized if one consumes healthy plant-based soluble fiber. So especially, how is cholesterol lowered by plant-based soluble fiber? Okay, this is what we need to understand, because each and every metabolic disease or metabolic condition is related with high levels of cholesterol. Okay, so we know that liver will produce bile salts to metabolize cholesterols. And once cholesterol is metabolized, bile salts are reabsorbed by the small intestine and they are cycled back to liver. Okay. So ultimately, cholesterol in liver is converted into bile salts. This is the normal metabolism. Now, what happens when one consumes plant-based soluble dietary fiber? So now the soluble fiber is going to bind with the bile salts. So this is going to prevent their reabsorption from the small intestine. So the bile salts are not available back again to the liver. So liver will be deficient on bile salts. So liver has to produce more bile salts. Producing more bile salts, that means decreasing cholesterol effectively, because new bile salts will be produced. Usually the bile salts are recycled when one consumes a diet which is low on plant-based soluble dietary fiber. So this is how liver gets a chance to produce more bile salts and digest more cholesterol. Okay. And of course, this also prevents diseases associated with liver, especially fibrosis of liver, which is caused by cirrhosis. Cirrhosis is caused by indiscriminate consumption of alcohol. That is also called as alcohol abuse. Next question which I would like to discuss is, why do vitamins do not need enzymes for digestion? We know that any organic biomolecule would require enzyme for digestion, because we will break complex biomolecules into simple ones by the aid of enzymes. The body does so. Then why not vitamins? Vitamins are very small molecules. They need not be broken down, or they do not need to be presented as molecules having more surface area for any enzymes to act upon. They are readily absorbed. So they do not need enzymes for digestion. They do not need to be broken down because of their size. The next question which I would like to discuss is, first of all, can we grow a liver, a whole organ in a lab? Is it possible? And if it is possible, to what extent? And if we are successful in that, where would we use the liver? So a simple solution which comes to our mind is that liver can be used as a transplant organ. Okay, it can be used to transplant to the donor, and the donor can get a new liver. But then the first question is, can a full blown liver be synthesized in laboratory? The answer is no. Because full blown, full differentiated liver to its maximum size can only develop during the natural course of human development, because it needs in vivo condition, human body condition, to achieve its full size and full differentiation. That is full specialization, full shape. Okay. But can we grow liver in lab? The answer is yes. This was done in University of Pittsburgh, and scientists so far have managed to grow mini livers, and mini livers are called as organoids. Now, these are, as I mentioned, not fully differentiated, full size organs, no, but they are simply a system of bile ducts and blood sinuses, but they are very small in size. So, do they have any application in medicine? Yes. They were transplanted into rats, and it was understood that these organoids can be used to test medicines. They were also used to treat a damaged liver. A damaged liver, a liver which had fibrosis, can be made more functional because the organoids which have been transplanted would take up the function of the damaged liver. Now, how is this organoid made? So, at University of Pittsburgh, as I mentioned, human skin cells were reverse engineered, or were reverse differentiated into human stem cells, and human stem cells are totipotent. We know that means they have the capability to differentiate into almost any kind of cell. So human stem cells were allowed to differentiate into hepatocytes, and thereby, by animal tissue culture methods, a full blown, not a full blown liver, but organoid, a miniature liver mainly made up of bile ducts and blood sinusoids, was synthesized, and this was transplanted in rat, because rat, of course, will serve as a model animal for clinical trials and stuff. So yes, there are applications of organoids. It is used to treat damaged organs, as well as it is used to test even vaccine safety, or safety of certain medicines or drugs. Okay. So this is all what I'd like to discuss today. Hope you have enjoyed. And if you are interested in such content, please subscribe to my channel. Please visit the link in the description. I have written a book, Curious Corners in Biology, which is a compendium of all such questions from every nook and corner of the subject, curious questions from every corner of the subject. It's like a stroke which will help you to be inquisitive in the subject, and churn things out of the subject, and it simply will make you feel biology more interesting. So definitely subscribe to my channel. Please hit the bell icon if you like the content. Otherwise, we'll see you later with more questions in physiology of digestion, and then we will be moving on to uh, physiology of respiration. Some curious questions into this topic. See you. Thank you. Bye.